So we have got the green signal from our community, technical community, that we are now live on YouTube. So we will be going. Good morning, everyone. So on this auspicious revision, we have Dr. Jagat Rathod here, and we will be discussing on the world of micro, means on the magical nexus of microbes. You are all are aware about how the microbes are working and how they help us. So we have our with Jagat Rathod, who will be dealing with and interacting with you on world of microbes. Before going further, I would like to invite Satyarth Pandey to introduce our esteemed speaker for this introduction for the webinar. Thank you, Satyarth. The platform is yours.
Thank you. Thank you, Sominder. Hello, sir, again.
Good morning,
So,
everyone.
let quickly introduce you all to our today's speaker, Dr. Jagat Rathod. He is one of the luminaries in their field with profound dedication and relentless knowledge. So, in 2004, he completed his bachelor's in microbiology from Gujarat University. In 2006, he completed his master's in microbiology from Bangalore University, and in 2014, he completed his PhD from MSc. So his work experience is as follows. From 2006 to 2007, he worked as a project fellow in University Grant Commission India. From 2007 to 2009, he worked as a SRF and GRF at Department of Biotech. And from 2009 to 2014, he did his PhD from the MSc. In 2014, he worked as a DBT RA at IISc Bangalore, and from 2015 to 2016, he worked as an associate scientist at AEMIN Industries. And then later, from 2016 to 2022, he worked as a postdoc fellow at National Seng Kang University at Taiwan, and currently he is working as an assistant professor at GBU, Gujarat Biotechnological University. Welcome, sir. So his
Thank
area
you.
of expertise are molecular biology, microbiology, anaerobic bacteriology, proteomics, and biochemistry. And his research focuses on environmental biotechnology, microbes interaction, physiology, bioengineering. He also appreciates the importance of science communication and actively motivates people to pursue career in science and research. He is a talented, dynamic, and diligent person showing excellence in every sphere of knowledge. With this warm now request Sommelier to proceed further in the interview.
Thank you, Satyarth, for such a wonderful introduction of our speaker. We will start with. I'm starting with a very igniting question for you, sir. When you were of our age, so what motivated to you to pursue the field of research? Means when think as like you were in our age, like a student of 20 to 24, 25 years. So what you were thinking that how you will go for research and all? Can you please shed some light on this?
Uh, yeah, thanks, Amelia, for this very fundamental question that I think every researcher might have gone through once in his lifetime,
Yes.
that he would like to take up research as a career option, or would like to indulge into this fascinating journey at one point of time. And I would must say that I mean, there are certain deep roots which goes all the way back to the school days, when we are learning science, and then we take up biology, physics, chemistry, mathematics, and then there are certain subjects which will fascinate you. Beyond this, I had an opportunity to learn some of the engineering subject during my school days. So like in the 10th board exam also, we had like electrical, mechanical, and engineering drawing. So I was actually taking those subjects, and my school was offering those subjects, so which kind of fascinated me to understand the systems at the engineering level, and try to uh, you know brings me to some of the basic concepts of engineering at the school time. And then there was some very unusual event happened, and later on that I got fascinated in biology, and then I kind of steered my interest towards biology throughout, and get into BSc microbiology, masters, PhD, and I think one of the very fascinating question which drove me to take up microbiology was when we were in first years of BSc, we have zoology and botany and things that you can see, but microbes are the ones which you cannot see. I mean, most of the time in the school days also we have microscope dissections of various things, and we can see life at a different scale in the plant and also in animals. But I mean, during my school days I never had an opportunity to observe microbes. And I think this is a kind of curiosity driven quest that started pretty early. And I think since I hadn't uh, you know kind of had any visual connect with microbes, and I was like just understanding things from the photograph, I think I was really fascinated to understand this world, and that's drove me to take up microbiology. And then as I kind of understood various fundamentals, I think it kind of drew me towards some of the basic questions, life at a small scale and how things work in the nature. And then that's why probably drive me towards environmental microbiology and to understand how microbes behave in environment. So yeah, so this curiosity is something that is something that will definitely make you drive towards certain choices. And then I mean, if you're really interested to understand the concepts and the how things happen and what sort of interactions take place, I think these are some of the very basic and fundamental questions that you start asking and then try to answer that, and then it goes on and on. So I think it's a more of a journey of curiosity, I would say. Uh, yeah, thank you.
Yeah, thank you, sir, for such a wonderful answer for this, for the very first question. So, sir, yeah, sir, now you are also a PI and you might be mentoring some students as well. So,
Absolutely.
sir, in your life, who have been your key mentors and who have played key influencers as a key influencers in your research career? Can you please shed some more light on that?
Uh, yeah, I mean, definitely you have a very strong connect with your PhD supervisor, because you spend a lot of time, understanding research through their views and how they, take up certain questions and, develops your critical thinking. So definitely, as a hours I spend with my PhD supervisor, I kind of, really had a fascinating time understanding how you should be asking a real big question. And I think beyond that, this also defines a way of meticulously following up with the question how you will do certain set of experiments, what are your hypotheses, what sort of strategy and the experimental designs you will you know use to answer those questions. I think MSU Baroda as an institute, I must say that definitely it's a state university, have a very limited resources, but I think some of the great minds and the kind of ecosystem is there, which kind of drags you to a completely different level. So even you are kind of have a different background in different subjects, but you basically the systems make you drive towards higher levels across different disciplines, and you have a great crosstalks which motivates you to know more and more. So I think this is more of a, definitely mentor will have a direct connect, and will have a great amount of influence on the way you do research. But I think ecosystem is also equally important. Like even in my postdoc mentor, he was a great guy, and very very open minded and allowed me to try different things. So I think postdoc mentors and PhD mentors definitely they have a great role to play, and that's very critical because the way they think has a great amount of influence on how you take decisions. So I think freedom in the lab is one of the very important criteria. I think that will make student more curious, and it's motivates you, because uh, I think this is something very very critical for bringing some very constructive minds on the table.
Yeah. Yes, sir. So I know, yeah, and the really the guru word guru, our guru really they play a very critical role in our life, as it is said in the Veda, Guru Brahma, Guru Vishnu, Guru Deva Maheshwara. Yes, sir. So, sir, as we all are, yeah, I have just entered into PhD life. What I heard from seniors, एक बार आ जाओ अंदर 2-3 साल में तो पता चलेगा क्या है PhD.
Uh-huh.
So, yeah, so that only I'm asking. So, in the PhD life, you might have overcome got some challenges and well that your results are not coming, and you're thinking that ये काम नहीं करेगा. So, how did you overcome that phase?
Absolutely. So I think this is something very, and I mean, you cannot avoid this situation. Probably every one of us might have, come across some situation during PhD where things are so frustrating, and you are thinking that maybe this is not going to work out. And then I think it's very much there in every PhD tenure, that some point of time that you might even go to the level that maybe this is not working out, I should think something different, or maybe just think about something else. But I mean, this is I mean, it was very much there in my PhD also, that there were certain objectives which were not working out, and there were certain objectives which were kind of giving me a kind of a bit smoother run compared to the other one. So I think it is very important that you don't stop talking and discussing your failures with other people, because what I experienced, that there are certain experiments which you are constantly trying to work it out, but there are certain small minute tweaking that might be required. And then if you're communicating with your seniors, your colleagues, even with your mentor, I think there is a great chance that they might give you a very critical and very constructive ideas that will kind of give you a way out. uh, you know Definitely there are certain of experiments that needs a third person to kind of evaluate and give you some sort of logic that maybe this is what is not correct, and then try to maneuver towards a better way of handling this situation. Another factor is time. Sometimes you need to give time, because there are certain experiments which demands lot of trial and error, and they need optimization, so you need to spend over it. And if you just think that I did it twice or thrice and it's not working and I give up, I think the research is all about training. So training your mind, your hands, your experiments, everything is required. So with that, i had a I had a great amount of learning across my mentors, colleagues, and seniors. Seniors really helped me out in coming out from certain big issues. So I think you need to be communicating, and if you are sharing your failures, you are probably inviting some of the solutions also.
Yeah, yeah, it's really, yeah. Communication is the key for the PhD. I feel that if we network more, and so that we can get answers, because we cannot think a lot. So, sir, eh,
And one more thing that I would like to add here is that uh, for this to a person to able to be communicative about all this, I think the ecosystem also needs to be very conducive. So if you are surrounded by people who think in a very different negative way, then you are kind of refrain from sharing. And if it is too much of pressure of performing positively always, then I think this is not going to work. So research needs kind of as probably one of the Nobel laureates said, that it's a more of a marathon, it's
mhm.
not a sprint. So this is very very important that you keep on running and try to improve every step.
Yes, sir.
Thank you. Yeah.
Yeah, yeah. So, sir, what I feel research is about all about observations and critical thinking. So sometimes we continue to get that negative data, what we don't want, but we don't keenly observe that, and some beneficial results might have hidden in that. We try to neglect that. So what do you think? How in this to the early research fellows, what do you think on your that how better observation will play a crucial role in their PhD journey? So what do you think on it?
Yeah, this is a great question, and I think I learned how to come out of this at the level of postdoc. So one of my postdoc mentor kind of taught me that what is the importance of positive control. So when you're doing protein microarray chip, one of the very critical thing that we always look is my positive control working out or not. Okay, so same, it's probably applied in across different research experiments that I think what we need is a positive control. And if those positive controls are working out, definitely they will guide you to, work out for your test and your samples, because they if the controls positive controls are working, that means you're performing your protocol almost correct. And it's just the sample that adds some complexity, and if you can crack that thing, then the experiment will work. So if we are focusing constantly on always try to work out the positive controls, and it's a kind of a confidence building exercise also, because positive control will give you some clear results every time. So if you are thinking positively, I think all those negative issues will try to
Yeah.
settle down, and with time you will reach to your destination with all
Yes.
the correct results and replicates and everything.
Okay. So, sir, one thing is that, sir, in the PhD journey, we keep on reading articles and doing experiments. So, ultimate, but that is also important. Apart from that, the peace of mind is also important. Like you're doing some exercise or doing some parties with friends. So, sir, the
Absolutely.
next question is that. How did you balance your research with other and balance your research with other aspects of your professional and personal life as well?
I think this is very important because when you're doing research, that demands a lot of continuous involvement, and
Yeah.
then you're constantly try to work out your experiment. And if things are not working out, you lead to kind of a very frustrative situation. So one thing that was very important, that if you are kind of having some sports as a kind of every evening you might be playing some small game of volleyball or maybe a cricket. And during PhD, we used to play probably in Baroda with even some of the faculties used to join from biochemistry. So uh, I think this is something a very relaxing moment, and there you can discuss science. It's just like if you're definitely you're involved in research, so every time things are moving in your head that this is not working out, maybe I should try this, and there you try to communicate with your friends, and then they might give you some good suggestions. And definitely taking trips out of the lab is extremely important. So during my PhD, I had like two very interesting trips, one to Leh and one to Spiti. And that was a kind of where I kind of discuss some of the good research question, as well as my future aspect, and how I would like to take up life with myself. And it's very very important that you take a break with friends, with colleagues, and probably also you will have certain set of people who are socially connected, not directly linked with your research. So it's a kind of gateway to different topics, and trying to meet people from different race. So that's very important.
Yeah, yeah. Really, thank you, sir, for such a nice and wonderful answer. Yeah, and currently I am just a year old into my PhD journey, and I have just last night I enjoyed a great and nice party with my friends.
Yep,
Yeah,
yep. We used to get some snacks from the outside, and used to make bhel inside the lab. And kind of, it used to be a kind of very relaxing moment, because you are trying to decrease the temperature and all the issues in the lab, and trying to come to some jovial settings. So that's extremely important.
Yeah. So without further delay, sir, it would be really great if we dive into the your research work and what is your research work and all, how, and so that you can share with our audience and students so that they can get enlightened with your topic.
Right. So basically, in my lab, basically we are trying to understand microbes mineral interactions and organomineral interactions. So those mineral interactions happens across different scale, and it's constantly happening in nature. So the question is that, can we learn how they happen, how they initiate those interactions, how they sustain those interactions, what is the chemical biology and the crosstalks happens, what is the kind of the ultimate outcome of different processes. And if we understand these processes clearly at the molecular level, maybe what we think is that there is a possibility that we can bring in some biotechnological solutions from learning from those interactions. Apart from that, the other research which is happening in my lab is trying to integrate functional materials in biotechnology, such as biochar or certain photocatalysts such as TiO2 and carbon quantum dots, and trying to see if they can kind of be coupled for a hybrid, technology development. The third one is kind of, we are, it's a kind of a long run, but identification, characterization of novel elemental storage and delivery system. So we are trying to understand some of the biomolecules which can act as a elemental storage system, as well as some of the proteins or certain enzymes which can be of delivery importance. And the
Yeah.
last is basically from a microbiology aspect, that if we understand the physiology or ecophysiology of microbes in various interactions, definitely it is going to add on to various agro-environmental biotechnological solutions, and making certain processes more sustainable, as well as it's going to bring out a certain hybrid solution. So this is what is happening in my lab. And yeah, so basically we are kind of a cross domain and interdisciplinary in our research. It's extremely important that
Yeah.
we integrate biology, chemistry, and physical signatures, and try to understand the system more clearly. And we're always try to be open towards various learning aspects. Yeah.
Yeah. Really, it's a really intriguing research, the interdisciplinary approach and the use of quantum carbon nanotubes and all. So it would be really great, sir, if you can share your slides so that we can learn more about it.
Sure, sure. So I'll try to be quick, as you
Yes.
mentioned. And so let me, one second, share screen. Mm, So can you see?
Yeah. Yes, sir.
Okay, okay. Let me. So is it visible? It's full
Yeah,
screen
sir.
now.
No, it's not full screen yet.
Is it not
No,
now?
sir. Sir, can you share entire screen instead of a window? Then it would be great.
Yep. I'll try to. Entire screen. Yeah. Okay. Yep. So now it's full screen, right?
Yeah, you can. Yeah. Now it's full screen.
Yeah, so thank you, and I'll be try to run through these slides and try to show some of the interesting interactions that we are trying to study in my lab. So basically, this presentation where I'll be discussing about microbes mineral interaction and their chemical biology conversations, which are very critical to understand from the nature science point of view, as well as there are technological benefits that you can derive from. So this is again my introduction. I'm skipping this. Yeah, so minerals probably like they are basic naturally occurring solids. Just try to give basic understanding about their minerals, and mainly inorganic in nature, based on crystallinity. It could be amorphous or crystalline, could be of metallic or non-metallic form, and that minerals have a direct link to national economy. And these are some of the minerals that which are of uh, importance, like igneous, sedimentary, and metamorphic rocks. And till now, a lot of mineralogical studies used to happen from geophysical and geochemical conditioning point of view, but in last probably 2 decades, I would say a lot of biological conditioning processes are getting understood. And here you can see that this is a cross section of stromatolites, and you can see the weathering through various microbial community, and each community imparts different color over here. So this is the green layer, pink layer, crystalline layer, and black layer, and that gives you kind of a succession or a stratification of different microbes on calcium carbonate stromatolites over here. So one of the question that very basic is, where do we find microbial interactions in the world? And here I'm showing you three basic interaction. One is the stromatolites, which are the living fossils and the oldest living life forms on our planet. The other one is at the nanoscale, and it's a nano inclusion in magnetotactic and the purpose sulfur bacteria, which is shown over here. And probably for this, you don't need to go very far. And this is basically dental caries, which is a hydroxyapatite bleaching by step mutants. And so basically, these interactions happen direct and indirect way, and there are various mechanisms which are really important to understand if you really want to dig out and understand the mineral microbe interactions. So this slides highlights across the time travel. This is a mineral microbes co-evolution. So on your x-axis you have a time scale from Euarchean period to Cenozoic period, and you can see that how different minerals diversity emerged, and how life forms also emerge across the time scale. And this is a kind of a graph showing how different oxygenation events lead to the generation of oxygen in the atmosphere, and the kind of different microbial interactions that were evolved across time. And you can see that earlier you had anaerobic life when oxygen was not there, and then you have a great oxygenation event, and slowly the oxygen started coming in, and then you have a diverse minerals getting developed across the time scale. And you can also see how biological biodiversity has also emerged out across the time scale. So there is a direct link between microbes and minerals in the time scale manner. So another question that would be really critical to ask is, why microbes interact with the minerals? So uh, these are some of the points which highlight that why microbes need to interact with the mineral. Some of the minerals can act as a terminal electron or donor, could be source for various nutrient system, and then that supports the microbial growth by providing substratum. They enhances their survival under various stress, could be chemical or physical stress, and definitely it gives you a physical protection. And some of the minerals are basically uh, excellent pH buffer and toxin binders, so that helps the biology to thrive in the nature. If we talk about again the mechanism, so you have a direct interactions, then these are the indirect through electron shuttles, and this is one of the very interesting nanowire driven mineral interactions. So when you think about scale, they are basically at the macro scale, where you can see the various rocks near the offshore. You have algal mats on them, and then at the nanoscale over here, you have a geobacter where iron minerals are getting kind of coagulated, and basically you have a nanoscale interactions taking place. One of the very interesting question is that does mineral inspire biomolecules? So one of the thing that greigite, which is iron sulfide, has a very structural similarity to some of the FES cluster which is found in ferrodoxin. So this is some of the very fundamental questions that is still driving that how minerals could influence the structural behavior in the biomolecule, and how the biomolecules could be inspired through minerals structures. uh, if you see the bacteria and fungal system in the nature, at the nanoscale also, like EPS strands help the microbes to interact with the minerals, and this is a cross section of fungal hypha, and you can see that various minerals are kind of present and on the outer surface. This is one of the example of ectomycorrhizal fungi interacting with minerals in the soil system, and leading to various elemental delivery system for the plants. Uh, Again, like these are some of the various surface interactions, like how microbes will first condition the surface, and then use certain cellular structures to interact with the surface, and there are certain forces which helps them to interact with different minerals in the nature. And there are two major processes that we are really interested. One is the mineralization. So you have a soluble ions which are present in the solvent system, and can we identify certain microbial system at the whole cell or the molecular level, which has a link to nucleation and crystal growth, and that's lead to basically the biocrystallization. And the other process is more of a solubilization, and which is a demineralization or bioleaching. And this is from one of our study where we studied the subsurface microbes interacting with the one of the most abundant arsenic source minerals such as arsenopyrite, and leaching out some of the toxic elements into the aqueous system and leading to the toxicity. Then at the elemental level, at the cellular atlas level, this is the mobilization processes. I'm not going in the detail over here. And the other one is the immobilization, which is through biosorption, accumulation, bioprecipitation, and bioreduction, which will kind of lead to a concentration of various elements. And it has a very great deal of biotechnological applications, which are really important in current era. And so this is what my postdoc work was, mainly on arsenopyrite. And so I'm skipping this. We had like two bacterial system which were directly interacting with highly toxic minerals and leaching out. We also did the whole genome and try to understand the synteny presence of various arsenic metabolic genes, and try to do RNA-seq to understand what are the genes which are playing the actual role in the interaction. And one of the major questions which will drive the biotechnological application of microbe mineral interaction is, you need to understand the molecular determinants, genes and proteins. What is the molecular mechanism of interaction, how they are regulated, what is the specificity of that process, and the ecophysiology from the microbiology side, which will help you to develop various microbiological processes. So again, I'm skipping. This is a more of a redox, changes we observed in arsenic speciation, and uh, this is the leaching behavior and the resistance behavior of the Staphylococcus strains to this arsenic. And as I mentioned in the previous conversation, this is a more of a interdisciplinary research. So you have a lot of biology. Then this is more of a physical, mapping of various microcolonies on the surface of minerals, and then chemical fingerprinting using synchrotron FTR mapping. So you understand what are the functional groups playing role in the microbes mineral interactions. So this is another example of bioaccumulation. And one of the critical thing is that when you're looking at the speciation, if the ions are in the solid form, I mean, when the element is in the solid form, you need to use the synchrotron-driven XS analysis to understand their structure and the oxidation state. So yeah, I think, and as I mentioned, the biocrystallization. So if you have a microbial community which kind of precipitates various elements, they can be of a great importance from the environmental biotechnology potentials. Also, from the ecological point of view, there are certain minerals which are having heavy metals. So when they are leaching out, it leads to heavy metal accumulation in the ecological system, and how it influences the whole biodiversity is also very important to understand. And again, like this is one of the slides showing the cross interactions of microbes or biomolecules to various ions, and how they can lead to certain crystal structures as a minerals. And if we understand this interplay, then you can develop various biotechnological applications for environmental cleanup, lithium ion recovery, and then microbial electron donation to anode in the microbial fuel cells, and biomimetics, and also leading to some of the AMR issues where you can develop certain nano structures which could be complementing to the antimicrobial minerals. So this is one of the other topic that we are also interested, where we are bringing in the biochar, microbes, and mineral interactions together and try to solve some of the agro environmental issues. I'm not going in detail here, and these are some of the teams with whom I worked in Taiwan, and now I started my own lab at Gujarat Biotechnology University. So yeah, I'm looking forward to great amount of interactions with students across different universities. And with this, I welcome everyone to GBU. So basically, we have some of the expertise in the medical, industrial, animal biotechnology, environment, and plant biotechnology. And with the support of bioinformatics and marine, biotechnology, we try to develop various biotechnological sustainable solution for society. And with that, I conclude. Thank you very much. Yep, I'm
Yes,
open to
it
questions.
was it was a really very nice talk, and so sir, we have a few doubts. We have very good questions in that our YouTube channel. So first of all, the first question I would like to ask that how do specific interactions between microbial cells and mineral surfaces facilitate the crystallization process?
So there are certain surface proteins, such as S-layer proteins or EPS-driven non-specific interactions with the ions, and which lead to kind of super concentrated microsystems. And if the redox and the pH supports this crystallization process, then they can form various minerals. And uh, if the minerals are stable, they sustain. If they cannot, they resolubilize again. So this is a process which is happening constantly. And one of the very critical thing that is there is that there are certain elements which are there in the soil system, but some of the soil minerals, they interact with those elements and then try to immobilize them, and that's why certain elements are not plant available, so such as phosphorus. So this is some of the very critical questions that we also are trying to address and understand, and try to find some solutions, because in the arid alkaline soil system, phosphate is quite a challenge for plants. And if we can develop certain phosphate storage and delivery system, I think that it would be really big boost to the agricultural system and to Indian economy, because we import phosphate every year.
Yeah. So see, you said you work on arsenic also, that arsenic and microbial interactions as well. So as we know, West Bengal water table has a high contamination of arsenic. So
Correct.
how bioremediation of arsenic can be done using this? If yes, sir, I will. And if yes, it can be done. What the biomass which will result? It also I will have arsenic contamination as well. So how to avoid that?
Absolutely. I think, as you know, that these are basically elements, and you cannot degrade element. You can just change their redox and basically from a soluble form to insoluble form. So as you know that when you use microbial or any filtering technologies, uh, definitely there will be a kind of a highly concentrated mass that will be either a biomass, or a filtering mass will be kind of generated, which will be highly concentrated with arsenic. So one of the way that could happen is that you try to set up certain filtration and microbial systems which kind of scavenge out certain arsenic ions which are highly toxic, like arsenic 3 and arsenic 5, which is arsenite and arsenate. And and definitely they will generate a waste which is highly concentrated with arsenic. So you need to also deal with how you will dispose of this biomass. So it's a very critical question. Uh, as a geological point of view, what one thing that could be done very critically is that you try to find out certain water sources which are less arsenic containing and try to see if we can utilize those aquifers compared to the one which are highly toxic, and also see that how nutrient recycling or the if we you know impose various nutrient system availability in the subsurface environment, that will increase the microbial activity. So there is a huge chance that it will kind of lead to arsenic solubilization also. So if we understand those biogeochemical processes, maybe we need to restrict the solubilization process, and in a proper mineralogical way also there are certain way out, like there are certain materials and there are minerals which can be utilized to scavenge the arsenic out. So not only microbiology but even from the mineralogic point of view there are certain solutions available, but then it need to be done at the system level, and definitely it's a big issue. Basically there's a large land which is kind of contaminated, and different tables of aquifers are contaminated at different levels. So I think it it requires a very systematic approach to deal with it.
Yeah. Thank you, sir, for your answer. So next question I have is that how actually microbes help in the formation of the minerals?
Sorry,
How microbes help in the formation of minerals?
in the formation of minerals. So one of the very important example is stromatolites. So there are some non-cyanobacterial and cyanobacterial activities which are leading to the calcium carbonate and those kind of mineral formation. And this is a basically living fossil and has a great connect to carbon storage. So as the ocean acidification happens, so these stromatolite structures are get integrated, and you have a release of carbon. So this is going to be a big impact on our sustainability and the ocean system. So if you think from the enzyme point of view at the level of molecules, I think there are certain systems which lead to kind of creating certain pH, like urease and carbonic anhydrase systems. They lead to calcium carbonate formations. So this is basically a microbial driven mineral formations that are of great importance. So apart from that, there are certain biomolecules which allows the certain elements to get concentrated, and in a kind of conducive system which allows them to get concentrated, lead to the mineral formation. But some of them are non-specific, some of them are quite specific.
Yeah, thank you, sir, for your such a nice presentation and nice research work, and you are sharing with us as well. So, sir, as we are ending, or means we are moving to the end of our event, and so the final the last question which I want to ask you, but there are many early career fellows which are watching our webinar as well. So, being a great PI, what advice would you give to students and the early career fellows for pursuing their research work, and mostly for the BSc and MSc students who want to become the early budding scientists in the future?
I think the very first point that I would like to put forward, this is just a suggestion, is that you follow your passion. Extremely important, because um, when if you're not passionate about science, research, or even technology to a certain extent, then it will have a direct link to the amount of involvement time that you're going to spend on your learnings. It has a direct link to your research time in the lab also, because if you don't like doing what, if it is not driving you, then probably you are going to spend less time and you will not even put effort to resolve certain issues. So I think most important is you follow your passion, interest. That's extremely important. At the same time, you have to be very watchful about the time that is going on, because sometimes in research it goes on, and then there are certain stages in the life that demands certain decisions. So I think it's very important that it has to be balanced out. Another thing that I would say is that try to, if you are interested to understand certain question, then make sure that those questions are quite strong and more of a futuristic, because when you are starting to answer that question, probably down the line after 3 years, you need to say that how relevant this answer would be. So if they are really relevant and some of the very critical answer to the some of the big question, definitely you will have a weightage of that research will have a great weightage. So it's extremely important that the kind of questions you ask and how inclusive understanding you bring in, and then basically those understanding need to be kind of very relevant to that time in future after 2 years, 3 years, 4 years. So there, if they find those values, then definitely it has a importance. So I think these are the two suggestions that I would like to give. And there are certain distractions also take place during research. So maybe sometimes you think that maybe this job is more important, and you give up your research. And then so I had my transition from PhD and then getting into industry, and then I realized that there are certain things that I would like to learn. So I left the industry, went into the earth science in Taiwan for postdoc, and then there I had a flexibility of learning so many things. I used to collaborate with material science people, medical college, and then you learn and pick up all the systems that you are interested in, and then basically you come back and you start asking some of the very critical questions that you think that would be important for your lab to grow with. So I think this is very important. So at the BSc and MSc time, I think fundamentals are extremely important. If those are happening at a great level, then it will kind of lead to a great foundation for your research.
Yeah. Thank you, sir. And really, we all enjoyed having you here in our event. And I wish we would be further looking for some great collaboration and interactions with you offline, sometimes offline as well. So, sir, since we are ending our event, I would really thank you from both
Thank you,
the
everyone.
Synapse community as well, and to our audience as well, and to all our YouTube audience. I would like to request that please do like, share, and subscribe to our the SciComm Snaps channel, so that we can come up with some great, great more integrated talks with sir and other great PIs as well.
All the best to
Yeah,
everyone.
thank you so much, sir, for joining us and giving your research out and sharing your highlights here.
Yeah, all the
Yeah,
best.
okay. Thank you, sir.
Bye.
Bye. And lastly, the feedback form is here in the chat box, so that please do fill up so that you can get the certificate on your emails. So till then, think, grow, and share. Okay, goodbye, everyone.